gut act on complex carbohydrates like starch and convert it into simple sugars like glucose that are absorbed in the blood. Let's first start with an overview about this process of glycolysis. At the end of digestion of carbohydrates, we get simple monosaccharides like glucose, fructose, and galactose. Please note at this point of time we have not extracted any energy from these sugars and in fact use energy to digest and absorb them. These sugars then undergo glycolysis, which produces acetyl-CoA. A small amount of energy is also produced at this step. This acetyl-CoA is then used in another biochemical cycle known as the Krebs cycle or the citric acid cycle, which takes place inside the mitochondria of the cells. In this cycle, we extract almost all of the energy that is present in these sugars in the form of ATP, NADH, and FADH. ATPs are utilized directly for energy, whereas NADH and FADH are high energy molecules that are used in the next step, which is the electron transport chain, where the high energy electrons present in NADH and FADH are used to produce a lot more ATPs. You can check out the videos on Krebs cycle and electron transport chain on our channel to learn more details about them. In this video, we will look at the first phase of carbohydrate metabolism, that is glycolysis, in which glucose, galactose, and fructose are converted into pyruvate and then to acetyl-CoA through a series of 10 reactions, and how these reactions produce energy. Glycolysis takes place in almost every cell of the body. If you look at the term glycolysis, you can see that it is made up of two words, glyco and lysis. Glyco meaning glucose and lysis meaning breakdown. So glycolysis is a process in which we break down glucose. We begin with glucose and end with pyruvate or lactate, depending upon whether oxygen is available in the process or not. If oxygen is available, we call it aerobic glycolysis, and almost all of the glycolysis that takes place in humans is aerobic, except some situations in which anaerobic glycolysis takes place, which doesn't require oxygen. We'll discuss these situations a little bit later in this video. Now let's start with the actual process of glycolysis. So glucose is transported inside the cells by special protein channels in the membrane of these cells. These channels are of two types, the GLUT receptors and the SGLT transporters. There are different types of GLUT and SGLT channels which are present in different types of the cells in the body, and they behave differently, also because different types of cells have different requirements for glucose. So glucose is inside the cells. Now what next? Well, the next step is actually very simple and the first step of glycolysis. Glucose which has entered our cells can easily escape outside, and to prevent this we attach a phosphate group to this glucose molecule to make this a glucose 6-phosphate, since a phosphate group is being attached to this glucose at the 6th carbon. This phosphate is provided by ATP and it gets converted to ADP. This makes the glucose molecule highly polar, and as you know, polar molecules cannot cross the cell membranes easily, so glucose gets trapped inside the cells. This step is irreversible and is performed by the enzyme hexokinase. Hexo means 6, meaning that these enzymes act on 6 carbon sugars like glucose and galactose, and kinases are basically those enzymes that transfer a phosphate group from a high energy molecule to another molecule, which in this case is a phosphate group that is being transferred from ATP to glucose. Now the enzyme hexokinase has 4 subtypes named 1 to 4, and the number 4 is known as glucokinase. Number 1 to 3 are found in most of the cells in our body, but the 4th type of hexokinase, known as a glucokinase, is specially located in the liver cells and the beta cells of pancreas. The 4th type of hexokinase is different from the rest 3 in a sense that it doesn't get saturated easily, meaning it can keep working even if there is a large concentration of glucose in the blood. This is important because large amounts of glucose is released from the gut after a meal that directly goes in the liver from the portal vein. Presence of such an enzyme that doesn't get saturated easily allows our liver to prevent excess glucose to enter our systemic blood circulation. In the pancreas, the glucokinase is a part of glucose sensing mechanism present in the beta cells of pancreas. Okay, so this was the first step in glycolysis, attaching a phosphate group to the glucose molecule and preventing its escape from the cells. This reaction was catalyzed by the enzyme hexokinase, and this reaction also uses one molecule of ATP that donates its phosphate group to the glucose. The next step is also very simple, in which an enzyme isomerase converts glucose 6-phosphate into its isomer fructose 6-phosphate. This isomerase is also known as glucose phosphate isomerase, which catalyzes this isomerization reaction, which could be reversed under normal cell conditions. After the formation of fructose 6-phosphate comes a very important step, in which additional phosphate group is attached to the fructose 6-phosphate, making it a fructose 1,6-bisphosphate. Bis stands for 2, indicating that it has 2 phosphate groups now. This addition of another phosphate group is catalyzed by the enzyme phosphofructokinase 1. Again, kinases are those enzymes which transfer a phosphate group from one molecule to another, which in this case is a phosphate group being transferred from ATP to fructose 6-phosphate. The ATP is in turn converted to ADP. So note here that we are actually using energy in this reaction also. This reaction here is very important because of the following 2 reasons. This reaction is irreversible because it is energetically favorable in this direction only. It is also very important in the aspect that it helps in controlling the rate of glycolysis. When our energy stores are low, this increases the AMP levels in the cell. The raised AMP levels in the cell signal the enzyme phosphofructokinase 1 to increase the rate of reaction. And on the contrary, if energy stores are high, meaning we have lots of ATP in our cells, this conversely decreases the rate of reaction so that the reaction proceeds at the rate that is optimal. But there are many other factors that control the rate of this reaction. We will discuss more on the regulation of glycolysis at the end of this video. Now, most of you will be familiar with the ringed and the open structure of glucose. But for those of you who don't, here is an important side note. Up till now, I have denoted glucose with this. This is the ringed structure of glucose. The carbon atoms in this glucose molecule are numbered from 1 to 6, starting with carbon number 1, which is the carbon atom next to the functional group. Now, no need to go into the details of this, but if you open this ringed structure, you get the open chain form of glucose, which looks like this. Again, you can note the numbering on these carbon atoms. Now, for the sake of simplicity, we can just represent glucose by this linear structure, in which we omit the other groups like hydroxyl groups and hydrogen atoms. The carbon atoms in this linear structure will be numbered from 1 to 6 in the similar manner. This is just to simplify the concept of how phosphate groups are attached and removed as we progress through the pathway of glycolysis. Let's proceed further with glycolysis. So we have reached a point in glycolysis where glucose has entered the cell. We have attached one phosphate group to this glucose molecule at the 6th carbon, making it a glucose 6-phosphate. Then we converted it into an isomer known as fructose 6-phosphate, and after that we have attached another phosphate group at the carbon number 1, making it a fructose 1,6-bisphosphate. This phase of glycolysis is known as the energy investment phase because up till now we have just used energy in the form of adding 2 ATPs, here and here. After this begins the energy generation phase, where we actually produce energy in glycolysis. This second stage of glycolysis begins with the splitting of fructose 1,6-bisphosphate into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So a 6-carbon fructose is split into 2 molecules which have 3 carbons each. You can easily remember their names by the mnemonic GAP and DAP. Now this reaction is reversible and it is catalyzed by the enzyme aldolase. Since the reverse reaction of this step, that is the combination of glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, is a type of aldol condensation, this enzyme is known as aldolase. These compounds are isomers and can be readily converted into each other by an isomerase known as triose phosphate isomerase. Again, triose meaning it can act on sugars with 3 carbons only, and isomerase since it is converting one isomer into another. This interconversion is important as only glyceraldehyde 3-phosphate can proceed further with the reactions of glycolysis. So dihydroxyacetone phosphate is isomerized into glyceraldehyde 3-phosphate, which is on a direct pathway in glycolysis. This isomerization results in the net production of 2 molecules of glyceraldehyde 3-phosphate from the cleavage of 1 molecule of fructose 1,6-bisphosphate. The preceding steps in glycolysis have transformed 1 molecule of glucose into 2 molecules of glyceraldehyde 3-phosphate. We now come to a series of steps that harvests some of the energy contained in glyceraldehyde 3-phosphate. For the next reaction of glycolysis, we have the enzyme dehydrogenase, also known as glyceraldehyde 3-phosphate dehydrogenase. And to understand this reaction, you have to dig a little bit deeper into its mechanism. Now, as you might know that dehydrogenases are enzymes that catalyze oxidation reduction reactions, meaning these enzymes remove hydrogen, that is why dehydrogenase, and removal of hydrogen is known as oxidation. The hydrogen that they remove must be passed on to some other molecule, and that molecule gets reduced, and that is how the dehydrogenases catalyze the oxidation reduction reactions. In this reaction, the enzyme glyceraldehyde 3-phosphate dehydrogenase acts on glyceraldehyde 3-phosphate and removes the hydrogen atoms from the carbon number 1 and 2, which are then transferred to NAD to make the NADH. Since water is also involved in this reaction, an OH group gets attached to the carbon number 1. Now this intermediate has high free energy and it is very unstable. It loses its excess free energy and the phosphate group gets attached to this intermediate, making it a stabler bisphosphate known as 1,3-bisphosphoglycerate. Because 2 phosphate groups are attached to it, now the new one being at the first carbon. So this is how the glyceraldehyde 3-phosphate gets converted to 1,3-bisphosphoglycerate. Now coming back to the main pathway of glycolysis, we started with glucose, a 6-carbon compound, which is then converted to glucose 6-phosphate, which after a few reactions is converted to fructose 1,6-bisphosphate, still a 6-carbon compound. It is then cleaved into 2 molecules which are 3-carbon compounds GAP and DAP. Only GAP can proceed further with glycolysis, which is fine as the DAP can be easily isomerized to GAP by triose isomerase. So if you look at it carefully, we now have 2 molecules of GAP for every 1 molecule of glucose that we started with. GAP is acted upon by GAP dehydrogenase enzyme and converted into a bisphosphate known as 1,3-bisphosphoglycerate, which is a high energy molecule and is used in the next few reactions to derive some energy in the form of ATP. The conversion of glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate uses a molecule of inorganic phosphate that is being attached to the carbon number 1. And this reaction is also very important in a sense that NAD is being converted to NADH. This NADH will be further used to make more ATPs in the electron transport chain. Now since we have 2 molecules of glyceraldehyde 3-phosphate for every 1 molecule of glucose that enters glycolysis, 2 NADs are actually being reduced here, which generate 2 NADH. And here you can see the second phosphate group that is being attached to the glyceraldehyde 3-phosphate to make it a bisphosphoglycerate. In the next reaction, the high energy phosphate group that is attached to the carbon number 1 is used to make ATP. The enzyme that acts on this reaction is known as phosphoglycerate kinase. And as you know, kinases are enzymes that transfer phosphate groups from one molecule to another. Here, the phosphate group at the first position in the 1,3-bisphosphoglycerate gets transferred to a molecule of ADP, converting it into an ATP. The 1,3-bisphosphoglycerate is